Hello AP Bio. This is a video for Unit 1, Topics 4, 5, and 6. So I'm going to go ahead and get the PowerPoint started and then we will begin. Great. All right, so this is all about biological macromolecules and properties, structures, and functions, Unit 1, Topics 4, 5, and 6. Carbohydrates. Let's start with carbohydrates. Take a look at this slide. Carbohydrates you have heard of in your biology classes. Of course, you know that those are sugars. This includes sugars and polymers of sugars. If you take a look at the structure of a carbohydrate, you know that it's going to have a carbonyl group that we talked about before and a hydroxyl group. It's comprised of carbon, hydrogen, and oxygen. We call simple sugars monosaccharides, and if you have more than one sugar hooked together, we call those polysaccharides. The repeating unit of a carbohydrate is CH2O. Most common is glucose. Glucose is a lot of times polysaccharides are made of many different kinds of sugars. Glucose is the one that your body needs for cellular respiration. Most common is glucose. It can serve as a building block for an amino acid. And it can also serve as a building block for monomers for di and polysaccharides. So carbohydrates, very important. Disaccharides, the Di represents 2. So if you have 2 monosaccharides joined together by a covalent bond, you call that a disaccharide. You all know disaccharides. Sucrose is a great example. If you have glucose and fructose and you join those two with a covalent bond, and we talked about that yesterday, by removing water, a dehydration synthesis reaction, you can actually make a sucrose molecule by putting those two together. Monomers of sucrose, glucose, and fructose. Plants transfer carbohydrates from roots to leaves in the form of sucrose. So a polysaccharide then is a polymer with many sugars joined through dehydration reactions. We want to take a moment to talk about the difference between a storage type of polysaccharide and other kinds of saccharides. Storage polysaccharides, like starch, they are very large saccharides. And so these starches are really, it's a way to store a lot of energy within a molecule, and then it can be broken down slowly to release the glucose that's in there. So it allows plants to store excess glucose. Now, animals don't store excess glucose in starch molecules. We don't have starch molecules as animals. So what do animals do? Animals store extra glucose in the form of glycogen. So starch in plants, glycogen in animals. We can find glycogen in liver and muscle cells. That's where we store our glycogen. And glycogen can be broken down when you need more sugar, and it's broken down to release the energy and to release those glucose molecules. We can also note a structural polysaccharide. Structural polysaccharides are incredibly important, and we don't always talk about this. When we think of carbohydrates, we're always thinking of energy, and excess energy. But structural polysaccharides are very, very important. Cellulose is that tough substance that forms plant cell walls. You got to have that. If it weren't for cell walls, in plants, that's what gives their toughness and the ability, in part, to be able to withstand water traveling up so far through the cells of plants. Chitin is another one, forms exoskeleton of arthropods and things like crabs, shells, those exoskeletons. And again, those are made from polysaccharides. Now, those polysaccharides, we don't have an enzyme that breaks those down properly. So we can't digest them, cellulose and chitin like that, but there are organisms that can. So the structural polysaccharide is an incredibly important kind of a carbohydrate, and we don't always take time to mention that. But termites sure love cellulose, and they have a special bacteria in their gut that can digest those tough cellulose cell walls. They have bacteria that can actually, that have enzymes in their guts that can actually digest that substance. There are a couple of practice problems for that information, and so you'll find that in your homework assignment that goes with this particular Unit 1, Topic 4, 5, and 6. So you'll see those concept questions in a cloud assignment. I'll move on from there. Let's chat about proteins. So we had carbohydrates, now we're doing proteins. So proteins, we're really big protein factories. And so let's chat a little bit about that. In general, we can take a look at this diagram and we can see that an amino acid is the basic building block of a protein. It's sort of like the Lego, and you put many of those together and you get this big protein. When you have a couple of amino acid molecules hooked together, we say that they are hooked through a peptide bond. Now, you know from our discussion yesterday, that's done through a dehydration reaction, removing water between two amino acid molecules. That forms a peptide bond, though. It's a special name for when it's with amino acid molecules. And so we form a peptide bond. If you have a whole bunch of those, then you have a polypeptide. That's what we have here. Now, you call it a polypeptide, so when you're looking at a protein being made in a ribosome, as the ribosome reads the messenger RNA, it knows which amino acid molecules to call for. It starts to bond them together through these peptide bonds at the ribosome. And as it comes out of that ribosome, it looks like this string of polypeptides. Not even calling it a protein yet, because we really don't call it a protein until it starts to form its 3D shape. Once it has taken on its 3D shape from the interactions of those reactive groups and has that 3D component to it, then we say it's a protein. So a molecule consisting of polypeptides folded into a 3D shape is what we would define as a protein. We say that's the definition of a protein. We know a lot about enzymes and proteins today and how important they are in our bodies. We can't survive without them, and enzyme reactions, without those, so many reactions going on in our bodies all the time, and we depend on those proteins, those special proteins, enzymes, to make that happen. Proteins are composed of carbon, hydrogen, oxygen, nitrogen, and sulfur, and the shape of that protein is so important. It determines its function. If you take a look at amino acids, you know, we did discuss this a little bit in the last video as well, that you have 20 different amino acids. How do they change? They vary only here at this side chain. The rest of this amino group, this NHH, and the carboxyl group on the other end is going to be the same for all amino acids. This R group is what varies. So the unique side chains will help the folding of the protein into that 3D shape. So unique aspects of the amino acid are based on the side chain's physical and chemical properties. The side chains are all grouped as nonpolar, polar, or charged. And what does that have to, how does that work with the folding process? If it's nonpolar, it doesn't have charged sides, it's hydrophobic, which means it is afraid of water. It wants to get away from the water. So when your polypeptide chain is starting to fold into a 3D shape, if you have an amino acid that is nonpolar hydrophobic, it wants to be in the center of that 3D shape. It wants to get away from the water. And if you have an amino acid that is polar, it's hydrophilic. It's water loving. Hydro referring to water, philic, love, like a philanthropist loves people and gives money away. So a hydrophilic is water loving. And then a hydrophilic amino acid, when it starts to fold, that whole polypeptide chain, when it starts to fold up into a 3D shape, any of them that are polar or hydrophilic will tend to want to be on the outside of the 3D shape because it wants to be closer to the water that surrounds it. Charged and ionic amino acids are also hydrophilic, so they act the same as a polar. They tend to want to be on the outside. So that can explain, why it folds the way that it does. The side chains interact also, and that's going to play a role in how it folds into its 3D shape. So side chains interact, and it's an incredibly important. The way that they are charged and how they're beside each other will determine its shape, and the shape determines its function of the protein. So that's very important. To form a peptide bond, if you look at this example here, you will take what happens is the carboxyl group of one amino acid is positioned next to the amino group over here of another amino acid. And what we have is the hydrogen from the amino group and the OH from the carboxyl group are removed, and that is going to be water. So that's a dehydration reaction. And that bond that results in the oxygen that's left and bonding to the rest of the amino group, that is what we call a peptide bond. So we don't have special names for all the bonds for the other monomers that are joined together, but for the formation of a polypeptide, we do call those peptide bonds. Many amino acids that are linked by polypeptides is then called many amino acid molecules linked by peptide bonds equals a polypeptide. And a lot of times students will ask me, well, when does it become a protein then? Why are you calling it a polypeptide? It's a chain of amino acids all put together with peptide bonds. Why don't you just call it a protein? Until it actually has that 3D shape, you call it a polypeptide. Once it takes on its 3D form, then you call it a protein. Each polypeptide has a unique sequence of amino acid molecules and directionality. Each end is chemically unique. One end is a free amino group, and one end is a free carboxyl group. The sequence of the amino acid will determine the 3D shape. Remember, what's so important is that shape determines function. If there's anything that you get from this section, remember that shape determines function. So when a polypeptide then is released from a ribosome, the first thing that it will do is it will start to twist and fold because of some of the hydrogen bonds that are being formed between some of the because of the polypeptide backbone, and then also because of the interactions of the reactive groups that are on there. And so this twisting and folding will make it take its 3D form, its shape. And that is very important for its function. So we can see sometimes when the amino acid sequence is disturbed through a mutation or something, that the resulting protein is messed up. It can't fold the same way because it doesn't have the correct sequence of amino acids. Here's a quick think, pair, share. How is the unique sequence of amino acids determined for a polypeptide? How do we get that unique sequence? Take a moment to see if you can answer that. If you said it's the genes, which of course are segments of DNA, coding for those, you were right. So anything that could mess up the amino acid sequence that was coded for from genes in DNA, there are a lot of places in there. You could have some messed up DNA sequences, messed up gene. The amino acids could have had a mutation as far as how they were placed. A lot of places along the line could mess up that code for the ribosome to read, and the resulting protein could be messed up because of that and won't fold into the right shape. Well, what are some functions of proteins anyway? Why are they so important? Functions of proteins include antibodies. We know how important antibodies are, keeping us from our diseases. And just equally, if not even more important, are enzymes. Enzymes are proteins that carry out chemical reactions or assist in creating new molecules. Enzymes are so important because if you, one example, you build up hydrogen peroxide in your body all the time. Well, hydrogen peroxide will kill you if you get enough of it at a high enough concentration in your body. So it naturally breaks down. Hydrogen peroxide will naturally break down, but it's a very slow process. An enzyme is sort of like a tool. It will grab onto a specific molecule, let's say hydrogen peroxide, and it does, it kind of squeezes it together just a little bit, which lowers activation energy for it to do its thing, in this case, breaking it apart. And so the enzyme does that, and then hydrogen peroxide just becomes a little bit of water, and it is not dangerous anymore. It's just a little bit of hydrogen or a little bit of water and a little bit of oxygen. That's why it bubbles, if you notice. But the enzyme can speed that up thousands of times so that it won't build up in your body. So enzymes basically make reactions that are going to occur anyway happen faster and in thousands of times faster. And so they're very important. That's just one thing that I mentioned, one little enzymatic reaction. Your body's full of thousands of those reactions happening, excuse me, all the time. Proteins are also messengers. They transmit signals, for example, hormones. Structurally, they provide structure and support in our bodies. We think of that usually with proteins. They do so much more. They transport and they store things. So they bind to and carry small atoms and molecules through the body. They are very important for that. There are more too. Those are the main ones. Let's talk about the levels of protein structure. There is primary and secondary, tertiary, and quaternary, 4 levels of protein structure. The first level, the primary level, is basically a linear chain of amino acid molecules that you would see just kind of coming out of a ribosome, freshly read and made polypeptide chain, and that is the primary structure of this protein. It's just a linear chain of amino acids. If you go to the secondary, well, we'll first do that. That's determined through your genes, yes, because your genes then are basically what are read, and then messenger RNA is made from that, which then travels out of the nucleus to your ribosomes. Your ribosomes grab onto that, read it, and then that is what codes for all of those amino acids. It reads the genetic code and knows which ones to grab. So genes from DNA determine primary and secondary and tertiary forms of a protein. Secondary form, once that chain is released from the ribosome, it will immediately start to coil and fold due to the hydrogen bonding within that polypeptide backbone. It can fold one of 2 ways. It can fold back and forth like a fan, as you see here. And this is called beta pleated sheet. These hydrogen bonds, you can tell it's a hydrogen bond because of the dotted lines, or sometimes we use dots. That represents hydrogen bonds. The hydrogen bonds are forming between the polypeptide chains that are laying side by side. And so we can get what's called a beta pleated sheet formation. That's one kind of secondary structure. Another one is called an alpha helix. It's when you have hydrogen bonding between every 4th amino acid. And these dots that you see is what keeps it like that. If you take a pipe cleaner and wrap it around a pencil and take it off, that structure that you would have is like an alpha helix. So that's secondary structure. Tertiary structure is when you have it fold up into a 3D shape due to its interactions to those amino acid reactive groups. So that's where we were talking about the side chains. Those reactive groups are either hydrophobic or hydrophilic. That's going to make it want to get the ones that are hydrophobic away from the outside and fold in the center, and vice versa. And that is what eventually will give you that 3D shape. We're doing an amino acid protein folding activity in class, so we'll get more of that. You'll get to see more of that in that activity. The 3D folding due to those interactions in the tertiary level of protein structure are reinforced by hydrophobic interactions and disulfide bridges of the side chains. Disulfide bridges are bridges that are formed between sulfur and sulfur, and so those are found in the amino acid called cysteine. Cysteine is the one that will have the sulfurs, and that's where those disulfide bridges are formed. So you don't see those until you get to the tertiary level. The covalent bond that's formed between sulfur atoms of two cysteine monomers, again, that is the definition of a disulfide bridge. Now, quaternary structure is when you have two or more of those polypeptide chains that are linked together, and we do find them in some proteins. All four levels of a protein structure determine the protein's function. You need all of them. They're all important. There's some practice FRQs for you that you'll have in a homework assignment, so I'm going to let you work through those. Nucleic acids. Let's get to topic 6. Nucleic acid is incredibly important too. I guess they're all important, aren't they? Nucleic acid is a polymer made of nucleotide monomers. So the building block is a nucleotide. Incredibly important. Its function is to store, transmit, and express hereditary information. We're talking about DNA and RNA. DNA, its name is deoxyribonucleic acid, and RNA is ribonucleic acid. So if we tear it down like we did for the others, we have nucleotides. Those are the monomer form. Many nucleotides grouped together are what we call polynucleotides. And then ultimately you have a nucleic acid, DNA and RNA, and it gets very large. The 3 parts to a nucleotide, 3 parts to the building block, the monomer, are number 1, nitrogenous base, 2, a 5-carbon sugar called pentose, and 3, a phosphate group. In polynucleotides, each monomer only has 1 phosphate group. So here are some nitrogenous bases: adenine and guanine, uracil, thymine, cytosine. We know them better from biology class, you remember? We always called them A and G, C and T, and then uracil as well, we find in RNA. 2 types. There are pyrimidines and purines. The pyrimidines, 1 ring with 6 atoms, and the purines have the double rings. So cytosine, thymine, and uracil are all pyrimidines. They're 1 ring with 6 atoms. You'll find cytosine and thymine in DNA, but uracil you find in RNA only. Purines are 1 ring, so all of these up here are the purines. And I'm sorry, all of these up here, the pyrimidines, are 1 ring, and the purines are the ones that are down here. These have 1 ring with 6 atoms bonded to 1 ring with 5 atoms. So adenine and guanine are the purines, cytosine, thymine, and uracil are the pyrimidines. Adenine, guanine. The 5-carbon sugar, our sugar is bonded to the base. In DNA, we call that sugar deoxyribose. And in RNA, the sugar is called ribose. So how do they differ? They differ a little bit in their structure and function. They're very close, but they are slightly different. You can see it down here with that oxygen and ribose. So the nitrogenous base we talked about, the sugar, and then there's a phosphate group. A phosphate group is added to the 5' carbon of the sugar, which is attached to the base to form a nucleotide. Take a look at this and what does this mean, 5' end? The way this works is we count the sugars. So this is, or rather the carbon in the sugar. This is considered 1, carbon number 1. This one is carbon 2. This is 3. This is carbon 4. This is carbon 5. So the 5' carbon is the one that you start with the one that is on the nitrogenous base. You start numbering them that way along the bottom, and that is the 5th one. So a phosphate group is added to the 5' carbon of the sugar, which is attached to the base, to form a nucleotide. You can see it here as well. Nucleoside, a portion without a phosphate group. So when you take a bunch of nucleotides and hook them together, what you see here are polynucleotides. Phosphate groups link adjacent nucleotides. There is a name for that linkage. It's called a phosphodiester linkage. You will probably not see that on your AP exam. So if we take a look at just one side of, let's say, what we've got here should be DNA. We have these nitrogenous bases, we have a sugar, and then we have the phosphate group. And notice that the phosphate group hooked to the sugar, hooked to another phosphate, hooked to another sugar, hooked to a phosphate, hooked to a sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. This is all just a backbone. It's just really a place to hang the nitrogenous bases. The code of life in DNA and RNA is here in the nitrogenous bases. This is the important message. The rest of it is just a place to hang it. So we have the 5' phosphate end up here, the sugar phosphate backbone here, and the 3' hydroxyl end on this side. The sequence of the bases along the DNA or messenger RNA is unique for each gene. So the sequence of bases, remember that the DNA holds all of our code of life. It has all of the messages there. So usually when a protein's being made or something's being made, it just needs a little recipe from the big DNA recipe book. And the sequences of the bases along the DNA or messenger RNA, it is going to be unique for each gene because each gene's coding for something different. It does dictate the amino acid sequence. And so we just talked about how important that sequence is for making a protein fold into its proper shape. It dictates the 3D structure of a protein. The DNA has 2 polynucleotides, 2 strands, if you will. And when it is hooked together, it forms a double helix. We say that the strands are anti-parallel. If you actually read the A's, G's, C's, and T's, you'll notice that you have TCA, GCTG, and you'll have the other side actually running it's an anti-parallel in that it codes up with its like partner, if you will. T always is going to pair with A, C always pairs with G, and again, A with T, T with A, C with G, G with C. So when you look at the whole strand, whatever the code is going up this way, it's going to go the same way going the opposite way. They're anti-parallel. Cysteine binds to guanine and adenine binds to thymine. RNA is different in that it is a polynucleotide, but it's single-stranded. And it doesn't have any thymine, it only has uracil in place of the thymine. So it's variable in shape due to base pairing within RNA. Adenine bonds to uracil, cytosine bonds to guanine. So as far as concept check, you have several of those to take a look at. I'm going to let you go through those on your own. Let's get to some lipids. The lipids are a class of molecules that do not include true polymers. Generally, they're small in size. They often are not considered to be a macromolecule. Lipids are nonpolar, they're hydrophobic. They do the types of lipids that you can find. There are fats, there are phospholipids, there are steroids. Oops, I'm gonna go back there. And those three types of lipids are very important. And for biology, these phospholipids, that's what surrounds each and every one of our cells. So we're going to talk about that because the lipid portion of it's hydrophobic, but the phosphate part of that is actually hydrophilic. And that's what makes it orient itself the way it does so that it can surround each and every one of our cells, protecting us and compartmentalizing our cells so that we can have certain reactions going on in certain cells and different ones going on in different cells. Fats are composed of glycerol and fatty acid. A glycerol is classified as an alcohol. It has hydroxyl groups. Fatty acids are long carbon chains, carboxyl groups at one end. 3 fatty acids do join to a glycerol through an ester linkage. That's what it's called, an ester linkage. They bond between a hydroxyl and a carboxyl group. They're classified as saturated fatty acid or an unsaturated fatty acid. What's the difference? Definitely want to know the difference between these two. If you think about it for a moment, saturated versus unsaturated. What are we saturating with? Hydrogens. So if you have a bunch of carbons and you actually have double bonds around, then you're not saturated with hydrogen because you have some double bonds, which means you have room for more hydrogen. Saturated fatty acid has no double bonds between carbons in the carbon chain. So this means more hydrogen, it's saturated with hydrogen. An unsaturated fatty acid contains 1 or more double bonds. This is a phospholipid. It's a major component of cell membranes. 2 fatty acids attached to a glycerol and a phosphate make up a phospholipid. So check out the charges on the phosphate part of this. You can see the phosphate here has oxygens, and you see negative charges on these oxygens. They do have negative charges, which makes them hydrophilic in nature, which means they like to be near water. This part down here, the tails, made of saturated fatty acids. Here's an unsaturated fatty acid. Right here we see a double bond. These are hydrophobic tails. They like to stay in the center of a cell. They're on the inside of the membrane. They don't like water, so they want to get away from that watery outside environment. Now, we'll see how it's a double layer when we get to cell membranes, but for now, just remember, the head part, the phosphate part, is water-loving, hydrophilic, and the tail part is hydrophobic. When we take a look at these, we can see how they'll actually form 2 layers in water. The tails are hydrophobic, the heads are hydrophilic. And again, we'll learn more about that when we get to membranes. Another type of fat is a steroid. And oftentimes we know when we're looking at a steroid because the molecule kind of reminds us of looking at chicken wire. If you've ever seen chicken wire, it has this shape to it, these rings that are hooked together in this kind of a pattern. This is, when you see something like this, you know you're looking at a steroid, which is a type of a fat. Lipids have 4 fused rings. Unique groups attached to the ring will determine the type of steroid that you have. Testosterone is one example of a steroid. There are lots of different steroids. Putting it all together. Hopefully you can fill this chart out with some responses. I'm going to put these in here for you so that it can help you study. I would try to make sure I could do this if I were you. Carbohydrates then, difference between proteins, lipids, and nucleic acids. Carbohydrates have carbon, hydrogen, oxygen. Compare that to protein, compare that to lipids, and compare that to nucleic acids. Obviously, there's a difference here. Carbohydrates only have C, H, and O. The monomer subunit is a monosaccharide. Put many of them together, you get a polysaccharide. Proteins. Proteins have carbon, hydrogen, oxygen like carbohydrates, but add to it nitrogen and sulfur. If you want the subunit or the monomer form of a protein is called an amino acid. Many of them linked together as a polypeptide. Lipids, made of carbon, hydrogen, oxygen, just like a carbohydrate. It also has phosphorus for the phospholipids. The monomer form, if you have to name it, is a glycerol and a fatty acid. We really don't say that they have polymer forms. Not really a true polymer. Nucleic acids are made of carbon, hydrogen, oxygen, just like carbohydrate, nitrogen, just like a protein, and phosphorus, just like a lipid. Nucleotides are the subunit or the monomer form, and the polymer form are DNA and RNA. So that actually concludes the entire unit and topic 4, 5, and 6. It's a lot of information, but I wanted to try and have it all in one place. Hopefully that helps you with filling out some of your information, and send me an email if you need some more clarification, and we can try to do Google Chats if you'd like to do that as well. My office hours, remember, are 2 o'clock to 3 o'clock every day at the end of the day. And if you need help even after that, just let me know. We'll go ahead and find some way that we can answer some of those questions. Okay, well, hopefully that helps you, and I will talk to you guys tomorrow.